All right, we are moving on to the cell cycle and division. And the reason why I put this right after DNA replication is because DNA replicates right before a cell divides. And so I thought this was a really good place for it to go. And so your notes will say cell cycle and division. And in order to reproduce, most cells are going to divide at some point in their lifespan. You do have some cells that are non-dividing. Muscle cells and your brain cells, your neurons, are actually non-dividing, but most cells are going to divide at some point in their lifespan. And the process is going to vary depending on the cell type. So like your skin cells are going to divide a lot faster than your heart cells or something like that. So it just depends on the type. So two vocab words here, somatic cells and gametic cells. Both are going to be involved, obviously, in cell division. One of them, though, is specific for today, and that's somatic. One of them will be specific for meiosis, and those are the gametes. Normally, teachers will do mitosis and meiosis together, but I don't do that. I think mitosis and meiosis each have their specific place, and I don't think they belong together. I just don't. Students tend to get really confused when you put them together. So today we're only doing mitosis. Mitosis is the division of somatic cells. Meiosis is the division of gametic cells. Somatic cells are anything that are not sperm and egg. So if you're looking at a cell, ask yourself, is this a sperm or is this an egg? And if your answer is no, then it's a somatic cell. If it is a sperm or an egg, then it's a gametic cell, also known as a gamete. So our focus today is on just somatic cells. All right. For most cells, the majority of their lifespan is actually spent in what's called interphase. Interphase you can think of as kind of like an intermission, although there is a lot of this stuff that's happening during this time. But it's kind of called an intermission because no division is happening during interphase, and the cell is doing its job and performing its normal functions and all of that stuff. So if you look under the microscope, these are plant cells, and you can see that they are square-like. That's how I know that it's a plant cell, because it's square-like because of the cell wall. If it's an interphase, it's going to look kind of like this. Nothing's really happening. During interphase, DNA is going to take on the form of chromatin, and I've used that word before, so it's definitely a vocab word that's important. Chromatin, remember, is DNA wrapped around these proteins called histones. And what I'd like for you to do is underline this word nucleosome. Underline the word nucleosome. So let's kind of follow this picture down. So look at the picture. I'm going to follow it downward. This is DNA. DNA wrapped around histones is called chromatin. Each little unit of DNA wrapped around histones is called a nucleosome. So each little, you can think of it as like pearls on a necklace. Each little circle you see here is called a nucleosome. So the definition of a nucleosome is DNA wrapped around, and it involves 8 histones. 8 histones. I'm going to see who's watching the lecture, so write down 8 histones per nucleosome, and then I'm probably going to ask that on the quiz. So what this is showing you right here is just a section of a chromosome. And then what I really need you to get from this is that chromatin condenses to form chromosomes. So chromatin, by definition, is just DNA wrapped around histones. A chromosome, by definition, is condensed chromatin. All right, so hopefully you got that. Okay, what I'm going to need you to write down is the word karyotype. Karyotype, that's what you're looking at right now, is this thing called a karyotype. So let me spell it for you. Karyotype, a karyotype. A karyotype is an organized arrangement of all of the chromosomes within the nucleus of one cell of an organism. And I'm showing you the karyotype of a human. A human has a total of 46 chromosomes. You're like, but why is it only numbered to 22? It's numbered to 22 because you double each one. So chromosome number 1, so there's a lot of genes on chromosome number 1 that has to do with eyesight and lots of other things. Chromosome number 1 has 2. It's a pair, one from mom, one from dad. Same thing with the chromosome all the way through to number 22. So you have 44 just regular chromosomes, 22 types, but a total of 44 because there's 2 of each. And then you have 2 sex chromosomes. Sex chromosomes are going to determine your biological sex. So what am I looking at right here? Am I looking at a male or a female? If you were to guess, a male or a female, so think about your answer, this would actually be a male because males are XY, females are XX. So you're looking at a male karyotype. And so this is just showing you the arrangement of chromosomes. This is what chromosomes actually look like. All right, so cells in interphase can be identified under the microscope by looking for the nuclei that contain chromatin, as opposed to the more condensed worm-like chromosomes. So make sure you pay special attention right now because I'm going to ask you, I'm probably going to put this on your quiz, which one of these cells can you identify that's in interphase? It would be one like this. Which one has condensed chromatin? It's going to look like this. And then which one is actively dividing? It's going to be this one. All right. So the nucleolus is actually visible in interphase also. So this is a perfect interphase cell. There's no worm like chromatin isn't super condensed, and you can see the nucleolus. All right, look at this picture right here, and I'd like for you to notate, because you don't have color on your paper, you're going to need to notate this. Everything in beige is interphase. Everything in beige is interphase. Everything in peach is mitosis. All right, so make sure you notate that. Interphase, everything in beige, consists of 3 parts. G1, S, and G2. During G1, the cell is going to replicate the organelles and the cytosol so that there's enough for 2 functional cells. Not only is the new cell going to get a copy of the DNA, it's going to get a copy of all the organelles and some extra cytosol to go with it. All right, so that's a G1 phase. And it's happening right here, and it's about 8 or more hours. During the S phase, DNA is going to be completely duplicated. So what was our topic over yesterday? DNA replication. DNA replication takes about 6 to 8 hours. We're talking about just like in general, on average. 6 to 8 hours. DNA replication happens during the S phase. And then the G2 phase is additional growth, preparation for mitosis, protein synthesis, all of that stuff. All right, that's interphase. So you definitely need to know what happens during each phase of interphase, okay? And then G2 takes about 2 to 5 hours. It depends on the cell. I don't like that I'm giving you hours here because it totally depends on the cell. All right, so we're going to talk about mitosis. So we're going to talk about this peach area for the rest of the lecture. Mitosis is going to follow these very specific steps: prophase, metaphase, anaphase, telophase, cytokinesis. How can you remember those in order? Well, it's PMATC, PMATC, PMATC. That's how you can remember it. So mitosis is the part of the cell cycle where the nucleus physically divides into 2. So remember, organelles have already been duplicated. There's more cytosol. DNA has already been copied. Mitosis is literally just the physical separation of one cell into 2. All right, and then this is your picture over here of what that actually looks like. I'm going to go through each phase, the PMATC. I'm going to go through each one. The P stands for prophase. Prophase is when DNA starts to condense. So those little the worm-like picture, I feel like this is a good prophase picture. This is also a really good prophase picture over here. Okay, this is what it would look like under a microscope. It looks like little worms are starting to form. The nucleolus is going to disappear. Spindle fibers are going to start growing out of the centrioles. Remember, spindle fibers are microtubules growing out of the centrioles on either side of the cell. I'm going to show you a really good picture in just a minute. Then what you have with your supercondensed chromatin and the chromatin with its copy is the X that we always see. Like when we're talking about chromosomes, we see this X. This X represents only one of the two chromosomes for a certain type. So chromosome 1 has 2, right? But if the cell's about to divide, chromosome 1 will look like 2 X's because chromosome from mom will be with its copy. Chromosome from dad will be with its copy. And so the chromosomes with their copy, each one, so this whole thing is called a chromosome, but each copy is called a chromatid, and the chromatids are connected with this thing called a centromere. There's a lot of C words. So if you go back to this picture right here, you see this X? This X is just, these are both chromosome 1, one from mom, one from dad, but they're each with their copy. Okay, hopefully this gets kind of confusing. So just know that the chromatids are joined together by the centromere. Okay, next part is called metaphase. The M stands for metaphase. It begins as the chromosomes move and align themselves in a narrow central zone in the cell. So they're lining up. Chromosomes that are really condensed with their copies are lining up in the middle of the cell. And it looks kind of like this. So the spindles are going to grow out. They're going to connect to the chromatids, the chromosome with its copy, the chromatids. They can't move independently. So if you have a chromosome and its copy, you want each new cell to have each one. So you want this thing to split apart, right? You want DNA and its copy to split apart. So you have the spindle apparatus that grows out of the centriole and attaches to each chromatid, and eventually, not for lack of a better term, but just to simplify it, it pulls these two chromatids apart. All right. And so metaphase, if metaphase involves the chromosomes lining up in the middle and the spindle attaching to each chromatid, anaphase is going to be where those two chromatids are actually being pulled apart. Okay, and this is what anaphase is going to look like under the microscope. So the centromeres of each chromatid splits and the chromatids are being pulled apart. They're pulled to opposite ends of the spindle by the spindle apparatus. And so they're just being pulled apart. And then finally in telophase, well, that's not finally, but it's the end. So PMAT-C, so this is the T. With the chromosomes now fully divided, the nuclear membrane is going to reform during telophase. DNA is going to return to its uncoiled chromatin form. The nucleolus is going to become visible again. Okay, hopefully you got telophase written down there. Nucleolus is going to become visible again. Okay, the cell still hasn't completely divided though. Cytokinesis is where the cell is actually going to divide. So in PMAT-C, cytokinesis is the actual division. Telophase is where the new nucleus forms. New nucleus forms during telophase. I'd write that down. The new nucleus forms. Cytokinesis is where the cell literally divides. So the cytosol organelles and cell membrane are physically divided during cytokinesis. The cell returns to interphase at this point. I don't want to go too fast. That's why I keep going back to make sure you have it written down. All right. And then this is going to be the specific way that cytokinesis divides an animal cell. So with cytokinesis in an animal cell, you have microfilaments that are going to wrap around the membrane and literally pinch it apart. So now it's two cells. And so the microfilaments wrapped around right here, that's called a cleavage furrow, and then it gets pinched apart. In plant cells, that's a little bit different. There's not a cleavage furrow and there's not microfilaments pinching the cell apart. Because of the cell wall, a plant cell is going to keep this shape right here. It's going to continue to be a square. It can't be pinched apart. So vesicles, remember vesicles are just like little bubbles of membrane that carry things. They're going to contain cell wall material and they're going to come and they're going to line up and migrate to the middle here. And then the cell plate will eventually form. It will elongate and then those two cells are going to basically be split in half. So what I'd like for you to know that in an animal cell, cytokinesis is going to be the forming of a cleavage furrow with a pinching. And then in plant cells, cytokinesis is going to be the formation of a cell plate and then just a really simple division. And so this is a really cool picture right here of a time-lapse microscopy of an epithelial cell. Epithelial cells are like the cells that line your organs. And this is really cool because it's fluorescent, meaning they put fluorescent markers and so they can see it glow. And so here you can see the two opposite ends of the spindle, and then the chromatids lining up in the middle. So what phase would this be? PMATC. What phase would this be? It would be M. These would be metaphase, chromosomes lining up in the middle. Okay, and then the last thing it says is once cell division is complete, the daughter cells will re-enter interphase until receiving a signal to divide once again. So that is mitosis, and those are the steps of cell division. Tomorrow we will talk about cancer and how DNA mutations are going to mess up the regulation of the cell cycle and cause the cells to divide out of control. I will have talked to you, or I will talk to you in Zoom about the time-lapse video. Basically what you're going to do is you're going to take all of these steps of mitosis and a list of vocab words, and you're going to go through and make a time-lapse video of this process happening right here. But I have very, very specific vocab words that you have to include. So you're using Play-Doh, vocab words, any other tools that you want to make your time-lapse video more interesting, and you're going to make a video of the actual process of mitosis. Time-lapse videos are a lot of fun, I think. A ton of fun, and it's a new, if you haven't done one before, it's a new tool for you to use, and I hope you have a good time with it. All right, and I will talk to you guys later.